Now I will talk about the topic chemi-osmotic hypothesis. Chemi-osmotic hypothesis. This is the today topic. What is the chemi-osmotic hypothesis? The title name indication is chemical energy synthesis with the help of osmotic hypothesis process. What is the chemical energy? Chemical energy is NADPH plus H plus. NADPH plus H plus, ATP. These two are the chemical energy. The chemical energy is synthesized with the help of osmotic hypothesis. This is the process. This is the production. Okay. This is the name indication. Next, the exact location of chemi-osmotic hypothesis is thylakoid. What is the location of chemi-osmotic hypothesis? Thylakoid. Okay, thylakoid is very important location for the chemi-osmotic hypothesis. This is the location. What is application? Second one is the application. Application means application is what is the importance of chemi-osmotic hypothesis. What is the main application? Synthesis of the exact location of ATP. The exact location of ATP in photo cyclic phosphorylation and non-cyclic photophosphorylation. Application is the exact location and synthesis. Location and synthesis of ATP in cyclic and non-cyclic photophosphorylation. Cyclic, non-cyclic photophosphorylation. This is very important application in the chemi-osmotic hypothesis. Okay, the exact location and synthesis of ATP in non-cyclic and cyclic photophosphorylation explained by the process chemi-osmotic hypothesis. Okay, this is the location, this is the application. Third one is what is the main process? Third one is important. Coming to the process. First start with the important of two important complex. What are the complex? Photolysis complex. The process start with the photolysis complex. Second one is a NADP reductase complex. NADP reductase enzyme complex. Reductase is enzyme. Is one of the complex. Okay, these two complexes are very important for the chemi-osmotic hypothesis. This is the process wise. Okay, this is the introduction part of the chemi-osmotic hypothesis. Okay, what is the location? What is the application? What is the process? Okay, location wise thylakoid membrane. Application wise the exact synthesis of ATP in cyclic and non-cyclic photophosphorylation explained by the chemi-osmotic hypothesis. Third one, process. This process is start with the help of two important complex. First complex is photolysis complex. Second complex is NADP reductase complex. Okay, this is the introduction part of the chemi-osmotic hypothesis. So, coming to the process. Coming to the process. Coming to the process is very important in chemi-osmotic hypothesis. First, the exact location is thylakoid membrane. This is the thylakoid membrane. Outer thylakoid having two important membranes. First, the membrane thylakoid having two important membranes. Thylakoid having two important membranes. Okay, this is very important. This is the thylakoid. Thylakoid having a two important membranes, outer membrane, this is called outer membrane, this is called inner membrane. The center of the thylakoid is called lumen, okay. The outer surface is called stroma. Outside of the thylakoid is called stroma. This is called stroma. Okay, these are the parts of the thylakoid. These parts are very important for the exact synthesis of the chemical energy, that is NADPH plus H plus and ATP molecule. Okay, first two important photolysis complex are present in the inner and outer thylakoid membranes. First photolysis complex located in the inner thylakoid membrane. Second complex is NADP reductase. These two complexes are important for the chemiosmotic hypothesis for the synthesis of the ATP. Okay, first complex name is photolysis. This is called photolysis complex. What is the need of photolysis? What is the process? What is the need of photolysis? Photolysis is useful for the splitting of water molecule to generation of the 2 protons, 2 electrons, 1 nascent oxygen molecule. These are the products of the splitting of water molecule. This complex name is called photolysis. What is the photolysis located? Location of photolysis complex is inner thylakoid membrane. This is the inner thylakoid membrane. Okay, this is the inner thylakoid membrane. This is the outer thylakoid membrane. Okay, first this is the location of thylakoid photolysis complex. Next, coming to the non-cyclic photophosphorylation. What is the main location non-cyclic photophosphorylation? This is the in between outer and inner thylakoid membranes. Two photosystems are observed in the in between outer and inner thylakoid membranes. First photosystem is, what is the photosystem name? Photosystem 2. Second photosystem name is photosystem 1. Which absorption center of photosystem 2 is? Photosystem 2 absorption center how much? 680 nanometers. Okay. Photosystem 1 absorption center is 700 nanometers. Wavelength. This is the wavelength ratio. Next, coming to the process. First, sunlight fall on the photosystem 1. Okay. The sunlight falls on the photosystem 1. In this reaction center having photosystem 2, which absorption center is 680 nanometers. Once fall on the sunlight, a large number of photons observed by the reaction center and accessory pigments. These protons are enter into the reaction center area. In this reaction center, a pair of electrons are excited from the ground state to excited state. In this accepted by primary electron acceptor, which is transported to electron transport chain, which is having a three important carriers. What are the carriers? Plastoquinone, cytochrome. This is very important electron transporters. Third one is plastocyanin. Okay, these three important electron carriers, with the help of a pair of electrons transported from one carrier to another carrier. So, gradually pair of electrons transported from excited state to ground state. Through this transportation through electron carriers, at the center cytochrome is very important for the synthesis of ATP. So, the pair of electrons gradually transported from excited to ground state. During this process, a large amount of energy is released. At the center of cytochrome complex. In this, this energy is utilized for the transportation of the protons from the stroma to, this is called lumen. Okay. The transportation of the protons from stroma to lumen. The high concentration of proton transported from the stroma to low concentration of protons in the lumen. So, what is the main reason? The proton are transported from the stroma to lumen with the help of energy at the center cytochrome complex. During electron transportation, some amount of energy is released at the cytochrome center. That energy is utilized for the pumping of protons from the stroma to lumen. The high concentration of protons which is transported into the lumen of the thylakoid. So, the proton concentration is increased in the lumen of the thylakoid. Okay, this is very important step in this ATP synthesis. Next, coming to the next continue processes, the pair of electrons is gradually enter into the photosystem 1. In this photosystem 1, again the reaction center, what is the absorption center is 700 nanometers. Once again the light fall on the reaction center, photosystem 1 absorbs the protons at the reaction center. In this reaction center, a large amount of energy is released with the help of photons. Large amount of energy is released with the help of photons which are released from sunlight. Okay, this energy are utilized further. Again a pair of electrons is excited from the ground state to excited state, again accepted by primary electron acceptor. So, the primary electron acceptor having a two pair of electrons, these electrons again gradually come down into the ground state with the help of another electron transporter called ferredoxin. So, this ferredoxin is important in the photosystem 1. Photosystem 2 having a three important electron carriers. So, these carriers are called plastoquinone, cytochrome and plastocyanin. These three carriers, cytochrome is exact location of the energy is released. So, the energy is utilized for the uptake of protons from the stroma to lumen. So, one of the cause is protons concentration increased from the, what is the source the proton concentration is increased in the lumen? First source is pumping of protons from the stroma to lumen at the center cytochrome. So, the cytochrome center is very important for the synthesis of the ATP generation. This is a one type of the mechanism. Second mechanism, the pair of electrons, second one is the photolysis complex. In this photolysis, the splitting of water molecule, a pair of electrons is continuously come down into the electron gradient. So, the replacement of the electrons from the photolysis complex. These electrons is again enter into the reaction center. So, the pair of electrons is enter into the PS1, a pair of electrons replaced with the help of photolysis complex. So, the electrons are very utilized from the photolysis complex. So, another important proton concentration source is splitting of water molecule. So, the proton are concentration one source is at cytochrome complex. Second proton source is from the splitting of water molecule. So, gradually the proton concentration is increased in the lumen of the thylakoid. So, the high concentration of proton is very important for the transportation of the protons into the, from the lumen to again come back into the stroma. Okay, next third byproduct is molecular oxygen. So, the byproduct of splitting of water molecule, a pair of electrons is replaced in the photosystem 2, non-cyclic photophosphorylation. Second product is protons. These protons increased concentration in the lumen. Third one is the oxygen, nascent oxygen is a byproduct of the splitting of water molecule enter into the stroma. The oxygen molecule which are nascent oxygen is enter into the stroma. So, these three are very important byproducts in the photolysis complex. So, first coming to the proton gradient. So, this proton gradient having a high concentration of protons increased in the lumen of the thylakoid, which facilitate the transportation of the protons into the stroma with the help of two important two important proteins. These proteins are two important proteins. First protein name is F1. Second protein name is F0. Second protein name is F1. So the proteins are these carriers. So these important carriers. What is the application of these two carriers? Transportation of protons from the lumen to stroma. The high concentration of proton which facilitate the transportation of protons into the stroma through the F0, F1 particles. The F0 particles which help in the facilitated by the protons, transportation of protons with high energy molecules. Two protons are transported into the F0 to F1 particles. F0 are very important for the transportation of protons. This F0 act as a carrier protein. What is application of carrier protein? Transportation of protons with utilization of high amount of energy. So the gradually enter into the F1 particles. F1 act as a ATPase. What is the ATPase synthesis? What is the ATPase application? The synthesis of the ATP. So the adding of ADP. First what is the application of ATPase? ATPase means synthesis of ATP from the ADP and inorganic phosphate. So the release of ATP, high energy. So the high energy molecule is very important for the energy generation. ATP with the help of ATPase enzyme. Okay, the ATP is released. What are the important substrate? ADP and PI with the help of ATPase enzyme. So this F0 is a protruding outside of the thylakoid membrane. F0 particle is embedded into the thylakoid membrane. Okay, which is embedded into the thylakoid membrane, and F1 act as a protruding in the outside of the thylakoid membrane. So these two proteins are very important for the transportation of the two protons from the lumen to stroma. These two are very important carriers. Stroma. Okay, next coming to the protons continuously transportation gradually come back into the lumen. These protons are very important for the energy generation. Okay, the protons is very important concentration gradient. This is the F0. So therefore ATP molecule and ADP. Okay, what is the application of two carrier proteins? The protons. With the help of the protons are transported from the lumen to stroma. Okay, the protons are from the high concentration to low concentration. This is called stroma. With the help of two carrier proteins, first carrier protein name is F0 particles, which help in the facilitation of the transportation of protons from the lumen to through the F0 particles with the help of high energy molecule. This high energy is important for the synthesis of ATP from the ADP plus PI through the F1 particles. F1 act as a ATPase enzyme. This enzyme is utilized for the synthesis of ATP with the help of ADP and PI and help of energy. Through this process, two protons are coming to the stroma from the lumen, high concentration to low concentration. So this is a very important step for the synthesis of ATP molecule. So this protons application is the protons are gradually increased in the concentration within the stroma. These protons are utilized for the synthesis of NADP. So utilized for the synthesis of NADPH from the NADP. NADP is converted into NADPH plus H plus with the help of NADP reductase. This is called the NADP reductase enzyme. So what is the application of NADP reductase enzyme? Synthesis of NADPH. This is a reducing power. This is called reducing power. The synthesis of NADPH coming from the NADP to electrons. What is a donor of electrons? From the photosystem 1. From the ground state to excited state to electron gradient through the electron transporter. What is the electron transporter in photosystem 1? Ferredoxin. This ferredoxin is important for the transportation of electrons from the excited state to ground state. So these electrons are utilized for the synthesis of reducing power with the help of NADP and 2 protons. What is substrate for the synthesis of NADPH? 2 electrons, 2 protons and NADP with the help of NADP reductase enzyme. NADP reductase enzyme located in the outside of the thylakoid membrane. So these are very important for the synthesis of NADPH enzyme. NADPH reducing power. So the synthesis of chemical energy. What are the chemical energy? NADPH and ATP molecule. These two are chemical energy for the dark reaction. Within the stroma. So the chemical energy released in the stroma. What is the chemical energy? ATP molecule and NADPH plus H plus. These two are chemical energy with the help of what is the process? Osmotic hypothesis. Osmosis is an important process. That is called the protons are transported. It's a high concentration to low concentration through the 2 important carrier molecules, F0 particles and F1 particles. Okay, these two are the embedded proteins and finally synthesis of ATP and NADPH molecule. These two are important compounds for the dark reaction for the synthesis of glucose molecule. And third carrier is CO2. So the CO2, ATP and NADPH, these 3 are very important for the synthesis of the glucose molecule in the dark reaction. Okay, next this is a overall idea about the chemi-osmotic hypothesis. What is the chemi-osmotic hypothesis? The chemical energy is synthesized from the osmotic process with the help of osmotic hypothesis process. What is the chemical energy? NADPH plus H plus and ATP molecules. So how the ATP is released? How the NADPH released? With the help of a very important process. First process is non-cyclic photophosphorylation and second process is cyclic photophosphorylation. So during this electron transportation, the energy is released at the center cytochrome complex. In this energy is utilized for the pumping of protons from the stroma to lumen. So the cytochrome acts as a electron transporter and proton acceptor. So the proton are accepted into the lumen. The lumen concentration within the lumen, the proton concentration is gradually increased. Another source of protons is splitting of water molecules. So the protons are coming from the 2 important sources. First source is photolysis of water. Second source is from the stroma to lumen through the cytochrome complex. So the proton concentration is gradually increased in the lumen. This high concentration is facilitated to the coming back to transportation into the protons into the stroma with the help of 2 important carriers, F0 and F1 particles. So these protons are transported from the lumen to stroma. The protons again used for the synthesis of the reducing power with the help of NADP reductase enzyme. So this chemical energy is synthesized by the process chemi-osmotic hypothesis. This is the overall idea of the chemiosmotic hypothesis. Coming to the next topic is dark reaction. So next topic is dark reaction. So thank you students.